Okay, welcome back to the thrilling conclusion of chapter 26. Whoops, I'm on the wrong thing. Sorry, there we go. Ha. Okay, let's talk about reproduction in Protists because they got a lot of different situations going on here. We have both asexual forms of reproduction in Protists and sexual forms of reproduction. So I want to talk about some of these asexual processes first. So they can do sort of just regular old mitosis, which is the equal splitting of a nucleus into two, and then it's followed by cytokinesis, so you make two separate cells from where one was. And that's one way they can reproduce asexually. Or, like we saw in the plasmodial slime molds, sometimes you can get mitosis that is not followed by cytokinesis. So you end up with this single cell that has multiple nuclei, so you get this large multinucleate organism. So those are two possibilities of asexual processes. We also can get something called budding as a way to reproduce asexually. Budding is when you have an outgrowth of a new cell from the surface of another. So if this was one of my cells, it might literally just start to bud off a new individual, and over time this individual would actually completely separate and become its own independent cell. And then we can also get something called sporulation. So sporulation is when we make specialized spores that are capable of just germinating into a new individual without having to fuse with anything. So we saw this in the cellular slime molds, where they would make a sporangium and they would make spores, and those spores would germinate, and then the individual cell that came out was good to go. No fusion, no sex, nothing, just it was happy being, just how it was. So that would be an example of sporulation. So those are a few different asexual processes we run across. Let's talk about some sexual processes. Because one of the interesting things we see, and we're looking at an example here, is that sometimes we get sex separated from reproduction. I don't know, that sounds kind of weird at first. But let's look at the paramecium as an example. So here's a paramecium. And what's going on in the paramecium is that we have two cells will align for something called conjugation. Okay, that term should sound familiar from bacteria. And they will swap DNA by the movement of micronuclei. The swapped micronuclei are then going to fuse with originals to form a recombinant macronucleus. So let's look at this image. So here we have some paramecium, and these paramecium typically have multiple of what we call these micronuclei. So let's look at what's going on. We have two paramecium align in conjugation, and what's going to happen is that in each cell, most of the micronuclei are going to disintegrate, except for one. But the one that remains, hold on, let me switch so you can actually, let me get my pointer. Okay, so most will disintegrate, but the one that remains will undergo mitosis to make many copies of itself. Then they're going to swap. They're going to swap around micronuclei so that each paramecium ends up with two distinct, genetically distinct micronuclei. One that they sort of originally had, an existing micronuclei, and one that was donated by the other paramecium. What's going to happen then is that these micronuclei are going to fuse, and then they will divide mitotically, and those will then sort of fuse together to make a new macronucleus. So where you see the purple, it's kind of this idea, red and blue makes purple. These two micronuclei have fused together. Now, I don't want you to necessarily worry about the details of each step here. But the concept that's really important here is that we are doing sexual reproduction, aka kind of the recombination of genetic material, without actually making a new cell yet. So that's what I meant by when I said that we separate sex from reproduction. So what's going to happen once we have this, once sex has occurred, we have this recombination, then we can reproduce. So after we've done this micronuclei swapping to generate some genetic recombination, the cells will disengage, so they won't be in this conjugation format anymore. And independently reproduce through a process called asexual transverse fission. So this is going to be like doing a round of mitosis and cytokinesis. And I don't have a good picture of transverse fission. Okay, so here we go. I found a couple of pictures. So this transverse fission, so what's happening here is the macronucleus is going to divide mitotically, and then we're going to get the paramecium actually sort of dividing itself asexually and making daughter paramecia. And so here is, you'll actually look at a figure in lab. Let me get my pointer back. You're going to look at a slide in lab that is going to be very, very much like that. Ah, sorry, I'm having technical difficulties. It won't let me have my pen. Oh, because I'm not in slideshow view. Sorry. Here we go. Let's try that. Okay, so here we go. So here's what you're going to look at when we go to lab of a paramecium in this transverse fission mode. And so this is purely asexual, but prior to this it undergoes the conjugation and the swapping of macronuclei to do its sexual reproduction. Okay, so now we're going to talk about something that is going to be a part of our lives for a while. And this is called alternation of generations. I'm going to hit on this idea when we get to plants as well, because this is something plants do, but I want to introduce this idea here because we see this in some produce. So this is a new, did I say produce? I meant protists, excuse me. This is a new kind of sexual life cycle that we haven't really talked about yet. So this is the succession of multicellular haploid and diploid phases. In some especially plants, big time in plants, but it's also, the reason we're talking about it here, it's also present in some algae. So let's look at this alternation of generations. Okay, so here's a life cycle, and we have two generations here. What we are calling the sporophyte generation and what we are calling the gametophyte generation. In this figure, blue is telling us haploid, so one set of chromosomes, and orange is telling us diploid, so two sets of chromosomes. The thing about alternation of generations that is different than some other life cycles that we've seen so far is that we have a multicellular haploid organism. So that's kind of different. And in some algae and plants, this multicellular haploid organism is actually really big, and sometimes it's really, really little. So the basic idea here, and we'll just start here at fertilization. So let's say two gametes have formed in fertilization and create a zygote. So a zygote, by the way, is just that first single cell that develops when egg and sperm unite in fertilization. Through the process of mitosis, this zygote turns into a multicellular organism called the sporophyte. So sporophyte means spore plant. So the spore plant produces spores by meiosis. And once we have done meiosis, we then are back to a haploid state, what we would call the gametophyte generation. Now, the thing about the gametophyte generation in this alternation of generations is that it develops into a multicellular organism. We're not used to thinking of it like that, right? Our egg and sperm never become multicellular. So like imagine a sperm able to divide by mitosis and become a multicellular organism that was the same size as you. That would be scary. But this is kind of what some organisms do, sort of. They have these multicellular haploid parts of their life cycle. And these gametophytes are what are capable of producing gametes. And gametes can fuse together and form a zygote and become diploid, and the cycle goes around again. We are going to talk about alternation of generations a lot in this class. So if this is a little murky, let me know. And we're going to, in the next few videos, I'm going to talk about it. And I'm going to talk about it in the context of particular groups. So hopefully it'll make a little bit more sense. But for now, just kind of get comfortable with this idea of a sporophyte and a gametophyte generation, and that they can both be multicellular. So finally, we're going to talk a little bit about some ecological roles of fungi. I'm sorry, of protists. I don't know why I have fungi on my brain today. Ecological roles of protists. So they can be very important primary producers, especially in aquatic systems. So they provide a food chain base, so they can be the base of the food chain. And they're very important in producing oxygen. they actually supply oxygen and the food base to many, many communities on Earth. Diatoms alone produce a fifth of all of the oxygen. And protists, when considered together, produce a third of the oxygen. So, yes, they're really, really important ecologically. Let me back up. Another important ecological role protists can fill is as a pathogen. And we might not like this one, but it's an important role nonetheless. So examples would be the Protist plasmodium, which can cause malaria, and the Protist trypanosoma, which causes sleeping sickness. There are, of course, many more than this, but these are a couple ones that affect humans that you may have heard about. Other human pathogens would include trichomonas, that would cause the STD trick, and many others. We'll talk about some in lab. And then finally, another ecological role is as an endosymbiont. So, for example, the radiolarians, which I talked about, have photosynthetic protists that live inside their shells a lot of times, and dinoflagellates are common in many animals as an endosymbiont. And when we get to animals, we're going to talk about a lot of them that have dinoflagellates as their endosymbiont. And they also can be found in other protists as well. All right, whoops, rogue pen. So here is just kind of a nice little summary table up there for you that kind of gives a nice view and a little image of some of these major groups that we've talked about. What a long, strange eukaryotic trip it's been. We're finally through the protists. You made it. Okay, so I will see you next time. Please let me know if you have questions. I know that this chapter is a bear, so please don't hesitate to talk to me. And we will also be looking at all these groups in lab, so hopefully this will give an opportunity to clarify points of confusion. So we will see you soon.